Hi, everyone. Just a quick presentation on the cultural characteristics of microorganisms. In other words, what are you looking for? Since bacteria were first cultured on solid medium, describing the appearance of the colonies has been an important tool for microbiologists. So what are you looking for? You're looking for the size of the colony. You're looking for any color or pigmentation of the colony. You're looking for form, which is the shape of the colony. You're looking at the margin, which is the edge of the colony. What does it look like? And the elevation, and that is how far has it grown up off of the agar surface. And so here it's just showing you some different pigments. Could be green or red or yellow or no pigment at all in the last one. Form is showing you the shape of the colony. Sometimes they're circular, sometimes they're just kind of irregular. Sometimes they look like they've got something growing out of the side of them. That's what they call rhizoid. The margin is the edge of the colony. What does it look like? The entire edge is nice and smooth, or undulate, meaning it's jagged, or lobed or lobate. Serrated, meaning it's like a knife, or filamentous, which means that it looks like lots of little filaments out on the edge. They don't look nice and smooth. And then elevation, you can have flat or raised or convex, which is domed, or ubanate, which is domed with a little bump in the middle. When we look at form, we're looking at a circular form in the first slide. The entire margin is nice and round, right, as serration marchans. And then we can have a regular form. There is Mycobacterium sphegmaticus, and you see that it doesn't look nice and round. And then finally, rhizoid form, you kind of look like you've got these little roots growing out of it. Here's some pictures of elevations, flat elevation, so when you look at it from the side, and a convex elevation, you've got that rounded dome shape, and then ubanate, where you've got that little bump in the middle of a dome. Here's some pictures of the margins. The entire margin is nice and smooth all the way around, undulate, lobate, and filamentous. So the description of a colony morphology or the cultural characteristics, this is what a microbiologist writes up. So it would give the name of the organism, and it would tell you what agar it was grown on, and it would tell you the size of the colony. So where do they get that size? They use a ruler. They use a little teeny tiny ruler. You actually have one in your microscope, in some microscopes where you can measure things as well. They lack, this would go on to say it lacks pigment. It does, and they're translucent, meaning they're see-through. The colonies are smooth and are circular in form with an entire margin. They have a convex elevation, and when manipulated with a needle, the colonies are viscous. So that would be the write-up we would expect for this particular organism. And this one's just showing you serration marchans. And you could do the same thing here, right? You could take a good look at this and you could write up a lot of information about the color and the size of the colony and all that sort of stuff as well. All right, so just some quick information there, some terms mostly for you to be aware of.